Hi dears, welcome back to botany session. And in these videos we are going to discuss about cell cycle and cell division. It is one of the chapter of 11th chapter, cell biology. And what is called cell cycle? It is one of the regular mechanism of cyclic events. Mechanism of the cyclic events or takes place in the cells. As a result, a cells ultimately divided into two daughter cells. These are called cell cycles. It was first described by Howard and Pelk in the year 1953. So Howard and Pelk, the two persons, first described about the cell cycle in the year 1953. It is a series of events. There are a lot of events that takes place in during cell cycle. What are the events? During cell cycle, the duplication of genomes also takes place within the cells, and all the cell organs also gets duplicated, and the result, a single parent cells are divided into two daughter cells. These are called cell cycles. But the durations between the two cell cycle, these are called generation times. Duration between the two cell cycle, these are called generation time, are also called the generation period. But this duration, it is varied from the organism to organism. It is not uniform. For example, in human cell, it will take 24 hours to divide into two daughter cells. But whereas in yeast cell, yeast it is a unicellular organism, it takes only 90 minutes divided into two daughter cells. So the period or durations of cell cycle, it is also varied from organism to organism. For example, in human cells, it will take 24 hours a single cell divided into two cells. But whereas in yeast cell, it will take 90 minutes to divide into two daughter cells. The next, this division of cell or cell cycle, cell division, it is one of the most important characteristic features of living cells. Most important characteristic features of living cell. Each and every living cells have the power to grow and divide. Grow, and they have the ability to divide into two daughter cells. It is the first most important characteristic features of living cells. The next, the new cells are formed from the pre-existing cells by cell divisions. Pre-existing cells by the process called cell division. It is nothing but it is proposed by Still and Schwartz. That's called cell theory. New cells. Next, increasing the number of cells. Increasing the number of cells by cell divisions. Cell division. And another one most important events of the living cell is the parent cell, they pass their genetic material into the daughter cells. Passing of genetic material such as DNA, because it is responsible for controlling of hereditary characters. The genetic material which is passed from parent cells to the daughter cells, from parents to daughter cells. So these are the important characteristics of living cells. Cell cycle, a cells will have the ability to grow and divide. Before growing and dividing, each and every cells they have prepared certain raw materials to divide. It's nothing but preparatory phase are also called interphase. So the before cell divisions, there are two processes are essentials. A single cell divided into daughter cells after the completion of two processes. One is nuclear division, another one is cytokinesis. The nuclear division is nothing but karyokinesis. Nuclear division, then followed by cytokinesis. Nuclear division is nothing but karyokinesis. The process of division of nucleus is called karyokinesis. It includes two stages. One is mitosis, then another one is meiosis. Then what is called mitosis? The mitosis is one of the cell division that occurs in the somatic cells. As a cell, this parent cell, the cytoplasm, it is a set of nucleus. The parent cell divided into two daughter cells. But these two daughter cells will have the same number of chromosomes as like in the parent cell. This is a parent cell. So here what will happen? The genome of the cell, it is nothing, or also total DNA content of the nucleus gets duplicated. So here duplication of DNA or also replication of DNA takes place not in the chromosome number. At the end of the mitosis, there is no change in the number, no change in the number of chromosome, but change in the structure of chromosomes. Structurally gets varied, but there is no number. If, for example, if the parent cells are diploid conditions, but all the two daughter cells also diploid number of chromosomes. These are called mitosis. Here only the genome of cells gets duplicated at the end. But where will it will happen? The mitosis cell division occurs when the cells to grow or it is replaced due to any damaged cells. The damaged cells or old cells are replaced by the new cells. At this time, this mitosis cell division will operate. Apart from that, certain organisms they asexually reproduce. Without formation of any gametes. The cells are divided into two daughter cells. For example, yeast. Yeast also reproduce asexually. At this time only, the mitosis cell division will operate. Once again, during the time of growing of the cells, replacement of the cells, asexual reproduction, then only the mitosis cell division occurs. But whereas in meiosis cell division, it is occurs in the reproductive part. But during meiosis cell division, the chromosome numbers are reduced to half. That is nothing but reduction division. See, it is a parent cell. It is a nucleus, cytoplasm of the cell, nucleus. See, the parent cell divided into two daughter cells. But these two daughter cells have haploid number of chromosomes. Parent cell have the diploid number of chromosomes. These two daughter cells have the haploid number of chromosomes. So these are called reduction divisions. So here reduction divisions are takes place. It is nothing but meiosis 1. So meiosis includes two division. One is meiosis 1, another one is meiosis 2. At the end of the meiosis 1, the parent cells, diploid set of condition of parent cells are divided into two daughter cells. These two daughter cells have the haploid set of chromosomes. Here what will happen? chromosome numbers are reduced to half from their parents. These processes are called reduction division. But second, meiosis 2 takes place in the daughter cells. It is a daughter cell. Here, meiosis 2 takes place. You know that in the meiosis 2, it is also called mitotic divisions. It is meiosis 2. In meiosis 2, there is no change in the chromosomal number. As like in the daughter cells are haploid cell, these two cells also haploid number of chromosome. It is also haploid. It is also haploid. It is also haploid. Already I point out, in mitosis, there is no change in the chromosome number. In meiosis 2, it is also like a mitosis. Hence, it is also called mitotic meiosis divisions or meiotic mitotic division. No change in the chromosome number. As a cell, 4 gametes are formed. 4 gametes are formed. So these are called meiosis. After the completion of meiosis, cytokinesis will operate. What is called cytokinesis? Division of cytoplasm. Only this mitosis, meiotic cell division is nothing but karyokinesis, only division of nucleus. Then followed by cytoplasmic divisions takes place. As a cell, single cells are divided into 2 daughter cells. These are called cell division. So the cell cycle, cell division takes place. Before cell division, cell cycle, all these living cells, they prepare itself certain raw materials, enzymes, DNA, RNA for protein synthesis. Everything are essential for the next stage. So before cell division, all these cells are prepare these raw materials. It is nothing but interphase are also called cell cycles. The next cell cycle. The next cell cycle. It is a regular events. A lot of events that takes place. A single cells are divided into 2 daughter cells. These are called cell cycle. The cell cycle includes 2 stages. One is interphase. Then second one is mitotic phase. It includes 2 stage or 2 phase, interphase and mitotic phase. Interphase, it is one of the longest phase. It takes nearly 95% of the total duration of the cell cycle. In total duration of cell cycle, 95% is occupied by interphase alone. It is also most active phase. But here the chromosomes are not clearly visible. It is appears like a thread-like structures. The chromosome appears like a thread-like structures, not clearly visible in this phase. Hence, it is also called a resting phase, also interphase. One of the most active phase, longest phase. It is also called non-dividing phase. Here there is no any divisions that takes place. It is a energetic phase, also called energy phase, because these phase prepare energy for the cell divisions. Hence, it is also called energetic phase or non-dividing phase. It is the longest phase. 95% of the total duration of the cell is occupied by this interphase. It is an active phase. Chromosomes are not clearly visible, hence it is also called resting phase. In addition to this, these are the most actively involved in the synthesis of protein phase. Synthesis of proteins. The protein that is essential for cell divisions. What are the proteins or energy ATP is essential for cell division? Everything they prepare itself in the interphase alone. Synthesis of proteins. This interphase, it includes 3 subphases. 95% by interphase, remaining 4 to 5% only take by mitotic phase alone. Now this interphase includes 3 subdivisions. 3 subphases. One is G1 phase. It is also called gap 1 phase or also called first growing phase. It is also called post-mitotic phase, pre-synthetic phase. Post-mitotic phase or also pre-synthetic phase, first gap phase or first growing phase, this G1 phase. The second one is S phase. It is also called synthetic phase. Synthetic phase. Here DNA gets replicated in this phase, S phase. The third one is G2 phase. It is also called post-synthetic phase or also pre-mitotic phase called G2 phase. It is a second gap phase or also second growing phase, G2 phase. So these are the 3 subphases which includes in the interphase. And it's a better easy to understand. I will draw the diagram here. Cell cycle. It is a mitotic phase. Mitotic, it is nothing but nuclear division phase. The next, it is a Z0 phase. Z1 phase. This S phase or synthetic phase. The next G2 phase or gap 2 phase. But these 3 phases G1, S phase, G2, it is nothing but interphase. It is an interphase. In the interphase, the G1 approximately 10 to 12 hours. It occurs 10 to 12 hours. S phase or synthetic phase 6 to 8 hours. G2 phase 2 to 4 hours. But whereas in mitotic phase 1 to 2 hours. But total durations, G1 phase occupied 42%. Totally interphase, it is a 95%. Total duration of the cells, interphase take 95%. Among the 95%, 42% take by this G1 phase alone. Remaining 33% by S phase, then 21% by this G2 phase. Next, only 4% by this mitotic phase. I will explain one by one. The first one is G1 phase. G1 phase. It is also called post-mitotic phase. I mean after mitosis. So it is a post-mitotic phase or also called pre-synthetic phase. Pre-synthetic phase. It is a first growing phase. First growing phase or gap 1 phase. These are called G1 phase. In this phase or the G1 phase, what are the material, enzymes, proteins are required for the next stages of cell division? Everything are synthesized in the G1 phase. I mean during this phase, synthesis of RNA, it is essential for protein synthesis. then synthesis of enzyme for DNA replication, because DNA replication takes place in S phase. Those enzymes are everything are synthesized in the G1 phase. Enzymes for DNA replication, then ATP synthesis, energy for cell division, ATP synthesis, then amino acid synthesis. In addition to that, nucleotide. DNA is made up of nucleotide. Such DNA gets duplicated in S phase. That is why what are the nucleotides essential for DNA gets duplication? Those nucleotides also synthesized in the G1 phase also. Apart from that, lipids, carbohydrate, then protein synthesis, protein. In addition to that, most of the cell organ, for example of mitochondria, then endoplasmic reticulum, everything gets duplicated in this phase. So it is one of the metabolically active phase, and they are actively involved with the synthesis of RNA, enzyme, amino acid, nucleotides, lipids, carbohydrate, proteins. Everything takes place in these cycles, in this phase. But in some cells are sometimes the G1 gets arrest at the middle or at the end. G1 gets arrest at the middle. That's called G0 phase. It is also the cells become differentiated. Differentiations. In some cell, the G1 arrests at the middle or at the end of the G1 phase. The cells gets arrest. That become growth and differentiation takes place. These are called Z0 phase. Then what is called Z0? It is also called quiescent center. Quiescent center or quiescent stage. Then why do we call it a quiescent stage? It's not a permanent stage. It's not a dormant stage. It's a temporary stage, normal functioning stage. Remember, in question G0, it is a metabolically active phase. Metabolically active, normal functioning also takes place in the Z0 phase. Why the Z0 will operate? Why the cells gets exit? Because this G1 phase, their failure to synthesis of any nucleotides, amino acids, ATP, enzyme for DNA synthesis, there gets failure to synthesis. Immediately, checkpoint will operate. These are called checkpoint. Checkpoint. What do you mean by checkpoint? It is controlled by cell cycle. Checkpoint, it is nothing but it is a mechanism which controls the cell cycle at the end of every stages. There are 3 checkpoints also there. Here checkpoint 1. Checkpoint 1. Here checkpoint 2. Checkpoint. Here checkpoint 3. Third checkpoint. At the end of each phases, checkpoint will operate. When will the checkpoint will operate? If the previous phase or G1 phase or S phase, their failure to synthesis of enzymes or amino acids, checkpoint will operate. Automatically, the G1 phase of the cells are exit. Cells are exit from the G1 phase, enter into the G2 phase for the normal functioning of the protein synthesis, DNA replication, everything that takes place. But it's not a permanent phase. It's a temporary phase alone. What will happen in the G1 phase? During G1 phase, this rate of RNA synthesis and rate of protein synthesis gets reduced temporarily. Rate of protein synthesis also gets reduced. But some cells, they are permanently existed in the Z0 phase. What are cells? For example, brain cells. Brain cells. Mature nerve cells are neuron, mature nerve cells are neuron, skeletal muscles cells. These cells are, they permanently exist in the Z0 phase. What do you mean? Here no dividing. These neurons are brain cells, nerve cells are, it's a non-dividing cells. They permanently existed in the Z0 phase. Hence it is also called quiescent stage or quiescent phase also. And checkpoint, if the checkpoint actually it is regulated by a special type of protein is called cyclin protein. Cyclin, it is a special type of protein. They activate the gene. When activate the gene, if the G1 phase gets deprived their work, they are failed to do their work, automatically cyclin proteins are gets activated to operate the checkpoint. If the checkpoint will operate it, automatically the cells are exit from the G1, enter the Z0 phase. There are 3 checkpoints also there. And if the checkpoint, suppose the cells are easily pass from the checkpoint to the next stage, these stages are, this phase is called a antiphase. These are called antiphase, are also called bullock. If they cross the checkpoint, their normal functioning will take place. Automatically cells are divided into 2 cells. Equal distribution of chromosome, equal distribution of amount of DNA also takes place after crossing the checkpoint. Hence it is also called antiphase. So in this phase also, cell size of the cell and the nucleus size also become double in G1 phase. The next S phase are also called synthetic phase. S phase and synthetic phase. During synthetic phase, DNA gets duplicated. Synthetic phase or S phase, it is nothing but a post G1 phase, pre G2 phase. That's called synthetic phase. It is also called S phase. It will take 33% of the total duration of the cell cycle, and it will occur minimum 6 to maximum 8 hours. Here replication of DNA takes place. Replication of DNA takes place. I mean, only the amount of the DNA become doubled. There is no chromosomal number, remember. Amount of DNA become double, but no chromosomal number. For example, in the S phase, 2C that become 4C. What is called C? C, it is a value for the picogram of the amount of the DNA. The C value, it is nothing but size of the it is a size of DNA in picogram. The size of the DNA in picogram, that is called C value. In haploid cells, C, it is a value for size of the DNA in picogram in haploid cells. In case of diploid cell, that's called 2C. So for example, the size of the DNA in the G1 phase is 2C. Amount of the DNA, only 2C. That it become 4C in the S phase. So in S phase, the 2C's amount of the DNA 2C that become 4C. So C, it is nothing but amount of DNA. But whereas in 2N, it is always equal to 2N. The N is nothing but number of chromosomes. So there is no change in the number of chromosomes during S phase. Chromosomes. Apart from that, centrioles. Centrioles, it is one of the cell division, cell organelle. It occurs only in the animal cell. The centrioles also gets duplicated in this space, along with these histone proteins. Histone proteins also synthesize or takes place in this space, that means S space. The histone, it is present only in the eukaryotic cells, not in the prokaryotic cell. Already I pointed out in previous classes, histone is nothing but a core of subunit of nucleosome. How the nucleosomes are formed? It is a core of its subunit of histone proteins. The DNA, they are identified approximately 2 meter of DNA is identified in these 2 meter of DNA, they are distributed in the 46 chromosomes. I mean the 23 pairs, total length of the DNA is identified 2 meter. How can possible this 2 meter of DNA, they are well packed or tightly packed or compactly packed in the chromosome because of these nucleosomes. This DNA, they are wrapped around the nucleosomes. So such proteins are called histone proteins. These histone proteins also synthesized in the S space, and the centrioles also gets duplicated in the S space. The next space, G2 phase. Next, G2 phase. The G2 phase, it is also called post S phase and pre-mitotic phase. It is a pre-mitotic phase and post-synthetic phase is called G2 phase. In G2 phase, it will occur minimum 2 to 4 hours and it will take 21% of the total duration of the cell is takes by this G2 phase alone. During this phase, synthesis of RNA and synthesis of proteins takes place. And in addition to that, a protein called tubulin proteins, tubulin and microtubules also synthesis takes place in this phase. Then what is called tubulin? What are the essence for tubulin protein? This tubulin protein is essential for the formation of spindle fibers. Spindle fibers. I mean the microtubules are organized to form spindle fibers. So the spindle fibers are essential for in the mitotic phase. They pull the chromosome towards opposite pole. Such spindle fibers in this takes place in the 3 phase, that is called G1 phase. It is activated by the protein is called tubulin proteins synthesis here. And in addition to that, cyclin proteins. So there are 3 types of proteins which regulate, which operate these cell cycles. First one is cyclin dependent protein kinase. It is a proteins that takes place or a synthesis in the G1 phase. What are the functions of this phase? Cyclin dependent protein kinase, it is a protein that will regulate the eukaryotic cell. Will regulate eukaryotic cell cycle is regulated by this protein. So cyclin, it is nothing but protein. It is a protein that activate the protein kinase. That activate protein kinase will regulate the eukaryotic cell cycle. Then another one is maturation promoting factors. The maturation promoting factor, it is a only protein, remember it is only protein that are synthesized in the G2 phase. Then what are the functions of this maturation promoting factor that regulate the transition of cell cycle from where to where? G2 to the mitotic phase. So this protein maturation promoting factor will regulate the cycle from G2 phase to the mitotic phase. It's carried out by this special type of protein. And the whole cell cycle is regulated by this protein. It's nothing but cyclin dependent protein kinase. It's a protein regulate the whole eukaryotic cell cycle. Apart from that, another one protein, the transition between the G1 to the S phase, G1 to the S phase, it is carried out by G1 cyclin plus CDC2 kinase. It is some proteins. So the transition of G1 phase to S phase is carried out by the protein is called G1 cyclin plus CDC2 kinase proteins. The transition of G2 to mitotic phase is carried out by the protein is called maturation promoting factors. Here G2 to mitotic phase factors. So after the completion of this G2 phase, interphase is over. Everything, all the materials for the mitotic phase. Cells, what other things are required for the nuclear division, those things, everything are synthesis in the interphase like G1 phase, S phase, G2 phase. Now the cells are divided in mitotic phase. It's nothing but nuclear phase. A nucleus is divided. It occurs 4 stages. One is prophase, metaphase, anaphase and telophase. Prophase, metaphase, anaphase, then telophase. After completion of telophase, the total amount of the DNA content in the nucleus gets divided, then followed by cytokinesis is nothing but division of cytoplasm. Cytokinesis, division of cytoplasm. As a cell, a single cells are divided into 2 cells. So how the mitotic divisions are takes place, how the nuclear divisions are takes place in this stage, we will discuss in another videos. Thank you.